The overall goal of this procedure is to generate cultures of human fetal retinal pigment epithelium, or RPE, that can be used as a model in investigations of human eye disease. Tissues are received from donors as intact eye globes. The first step of the procedure is to trim away extra muscle and connective tissue and then open the eye by removing the cornea and lens. The eye is treated with dispace and flattened, and then the RPE monolayer is peeled away from the choroid. Dissected RPE are seeded into flasks and grown for several weeks until they reach confluent monolayers and develop pigment. Cell cultures are used in vitro functional experiments. Successful in vitro experiments are repeated in preclinical trial animal models where RPE responses can be better evaluated in more complex signaling surroundings. The model demonstrated here will lead to better understanding of RPE physiology and new therapeutic interventions to treat ocular diseases.
Hello, my name is Sheldon Miller. My laboratory is located in the National Eye Institute, which is part of the National Institutes of Health on the Bethesda, Maryland campus.
Hi, I'm Arvidas Maminishkis from Miller Lab.
Today we will show you a procedure for preparing primary cultures of human fetal retinal pigment epithelial cells. These cells will be prepared as confluent monolayers that can be mounted in modified Ussing chambers. We will also use these isolated human RPE cells for studies in biochemistry and genetics.
We use this procedure in our laboratory to establish primary cultures of epithelial cells to study the function and regulation of physiological and pathophysiological processes and develop preclinical animal models of diseases. We focus on the retinal pigment epithelium, or RPE, which sits in the back of the eye, separating neural retina and choroidal blood supply.
This schematic diagram of the eye shows the location of the RPE sheet in the back of the eye just adjacent to the photoreceptors. The insert shows the cells of the retina with the photoreceptors on the right and the ganglion cells on the left. This epithelium is metabolically extremely active. It phagocytoses photoreceptors continuously on a diurnal basis, and it stores, reisomerizes, and shuttles the visual pigment vitamin A to the photoreceptors in the visual cycle. It acts homeostatically to maintain the chemical composition and volume of the surrounding extracellular spaces. A critical property of epithelia is their polarity. The ongoing asymmetric delivery of proteins to the apical and basolateral membranes is a regulated process that allows the epithelium to carry out the vectorial traffic of metabolites, ions, and fluid from one extracellular space to another. This activity helps epithelium maintain the health and integrity of the neural retina. The in vitro preparation that we describe here allows the analysis of transport proteins, receptors, and signaling pathways that determine epithelial polarity and function. Before beginning the dissection, prepare a 12-well plate and a laminar flow hood with room temperature solutions. Each eye requires 1 well of 10x antibiotic antimycotic solution, 2 wells of 1x PBS, and 1 well of 2 units per milliliter of dispace diluted in 5% RPE medium with serum. Fill a silicone padded dissecting dish with 1x HBSS. Drain the liquid and transfer each eye into 1 well of antibiotic antimycotic solution. Incubate for 3 to 5 minutes at room temperature. Avoid applying any excess pressure to the eyeball. Rinse the eye in 2 successive wells of 1x PBS to remove excess antibiotic. Then use forceps to transfer it to the dissecting dish filled with 1x HBSS. Under a dissecting stereo microscope, use iris scissors to remove excess muscle and connective tissue around the eye. Align the eye so that the cornea faces up. Use 2 sterile 27-gauge needles to pin the eye by connective tissue without damaging sclera to the silicone pad in this orientation. Use a side port knife to make an incision through the sclera. The cut should be 1/3 of the distance from the eye equator to the anterior surface. Use iris scissors to continue the cut circularly around the eye. To avoid damage to retinal pigment epithelium, abbreviated RPE, by vitreous retina traction, use the same scissors to cut through the vitreous body to separate front eye flap from eye cup. Lift the anterior portion of the eye away and discard it. Use forceps to transfer the open eye into the well of dyspace. Make sure there are no air bubbles in the eye cup. Incubate for 40 to 60 minutes at 37 degrees Celsius with 5% carbon dioxide. Transfer the eye from the dyspace back to the dissecting dish, which has been filled with fresh room temperature 1x HBSS. Position the eye with the cup facing up and secure it with two 27-gauge needles. Under the dissecting microscope, use fine forceps to gently lift the partially separated retina. With retinal scissors, cut the retina away from the optic nerve. Discard the retina. With the iris scissors, make an incision from the periphery of the eye to the optic nerve. Pin the eye out with 5 27-gauge needles to stretch the RPE layer. Use retinal scissors to make a circular cut around the optic nerve, separating the RPE layer. Switching to 250x magnification or more, find the edge of the RPE sheet along the cut made by the iris scissors and close to the optic nerve. Use 2 pairs of forceps to separate the RPE and Bruch's membrane from the choroidal tissue layer. Try several areas along the cut edge to find where the connection between the RPE and choroid is weakest. Gently peel the membrane back to remove it completely. Place the RPE sheets into a 15 ml conical tube filled with room temperature trypsin EDTA. When all the sheets are collected, cap the tube. Incubate the tubes in a water bath for 10 minutes at 37 degrees Celsius. Shake the tube vigorously to separate the RPE into small clusters. No dark cell clumps mean separation is complete. Return the tube to the water bath for another 5 minutes. Use a Pasteur pipette and gentle suction to remove any undigested tissue leftovers. Spin down the cells in a centrifuge at 285 G for 4 minutes. Remove the supernatant. Bring the final volume up to 9 ml with 15% RPE medium. Pipette to resuspend the cells. Pipette 3 ml of the cell suspension into a 25 cm squared flask. Add 2 ml of fresh room temperature 15% RPE medium. Culture the cells overnight at 37 degrees Celsius and 5% carbon dioxide. As the cultures mature, their pigmentation will increase. After 3 to 4 weeks, successful cultures will be confluent and uniformly intensely pigmented.
We have just shown you how to prepare human fetal RPE primary cultures. So that's it. Thanks for watching and good luck with your experiments.